everyone. This is a little bit different kind of presentation. So I'm talking about how to produce hydrogen, which is low carbon in nature, by using waste as a feedstock. So using biological route. So I'm from Indian Institute of Chemical Technology, the CSR lab. So we discussed. I'm not talking about that one. So first, coming to the biogenic hydrogen production, basically there are two diverse routes. One is with biomass. When we use biomass and thermochemically we convert it, the hydrogen which is coming, we can call it as biogenic, which is one of the domain which many are pursuing. Apart from, there is a complete biological process where we can use bacteria like anaerobic bacteria. By dark fermentation we can produce hydrogen, or using photosynthetic bacteria, by photosynthetic mechanism we can produce hydrogen. Apart from that, we can isolate the enzyme, hydrogenase enzyme, we can produce hydrogen, or with a mechanism, hybrid mechanism, using dark fermentation, by giving some input of energy, we can again produce hydrogen. So among these processes, I would like to say photosynthetic biologic production, theoretically it can produce more hydrogen, like 8 moles for 1 mole of carbon, but very difficult because of oxygen generation. So, however, with anaerobic process, even though it's a low conversion process like 4 moles per 1 mole of glucose, that is practically feasible and we can scale up with dark fermentation. So, we are working with dark fermentation process. We started looking into production in a lab scale at 2005, and then we came up to a pilot scale demonstration in 2016. And now we are moving towards demonstration at higher capacity. So I will go through briefly, not going into the deep, to explain how we come up to this stage. So the basic our objective is to use the mixed culture, which is very much similar to methane production. So methane production, there are 4 steps. So we want to cut the last step, where anaerobic bacteria can produce hydrogen as the terminal end product. So carbon in the form of glucose, when we look into 1 mole of glucose, we produce 4 moles of hydrogen when it goes to acetic acid pathway. But theoretically, 1 kg of COD, carbon load, when we are able to remove, we can produce 468 liters of hydrogen. This is the theoretical limit. So all wastewaters can produce hydrogen when we look into the wastewaters, but there are a lot of challenges. So some of the challenges we try to list here, but these are some of the critical things we try to address before going to the pilot scale. The one thing is, when we use the mixed bacteria, all kinds of bacteria will be there, mithrogenesis, esterogenesis, but our interest is to suppress the mithrogenesis in order to make hydrogen as a terminal end product. So we try to look into some of the strategies, classical microbial techniques, try to suppress mithrogenic bacteria, at the same time enrich estrogenic bacteria. This has worked us very well. So this will take our process near to the mithrogenesis, because we cannot use single cultures, even though single cultures are effective, but when we use wastewater, that cannot be feasible. So this strategy worked out well, and then we try to optimize the reactor conditions, how to immobilize the bacteria at extreme acidic conditions, and then loading rate. If more load of carbon we are giving, we are getting more of hydrogen. So but we cannot extend certain limits, because there is a formation of fatty acids. The bacteria get inhibited because of pH change. So we try to look into that aspects, and diversity, microbial diversity. Basically, firmicutes are known hydrogen producers. So we look into how this system is moving with diversity almost 3 to 4 years, and observed that firmicutes are persisting when we are operating system at pH 6.4. These are the optimum conditions, and based on that, we have set up a pilot plant with funding of Ministry of New and Renewable Energy, where TERI and IIT Kharagpur are also partners in this project. This is the pilot plant system with 10 meter cube capacity, with all axes to regulate the bacteria and other things. So the photograph view of different unit operations. The objective here is to use biogenic waste and to produce near to 50,000 liters of hydrogen per day. So we are able to achieve this one, but we have some problems. So that I will explain. So the basic problem is, even though we are getting hydrogen, the carbon which we are feeding into the system, 50% of carbon still remains in the reactor, which is coming from outside. So that is not a big deal. It's a big deal because we have to treat it before disposal. So that is one major problem. And then we try to look into valorization. So valorization means the carbon which is coming from reactor still it has value. So it's in the form of acetic acid, bitric acid, which are platform chemicals. So we look into those aspects and integrated with a different secondary biological process, wherein some of the important things are bioplastic production. The fatty acids which is coming from reactor will be feedstock to produce biopolymers, so that we can convert 90% of carbon to hydrogen as well as plastics, or with microalgae cultivation we can convert into biomass, or we can use it as a phosphate solubilizing agent, which can increase the productivity of plants, because mild acidic conditions will increase the phosphate availability. These integrations we try to look into. The advantage of this biorefinery is able to remove or able to use 80 to 90% of carbon which we are feeding into the system, apart from deriving two bio-based things. One is hydrogen and second one is some of the platform chemical. So these are plastics which I have discussed. These are the plant experiments which we have done. We got very good productivity when we are adding 10% of the outlet from the reactor. So as I told, then we focused on separating acids, acetic acid, bitric acid and propionic acid, which are also byproducts. So this has more market value than the hydrogen in a given system. So the basic idea now is hydrogen became the secondary product, but these platform chemicals now, which is being produced by fossils, became the major interest for us. So we try to increase the acetic acid or bitric acid, we are getting more of hydrogen. So based on these, another important thing is we try to look into production of biohydrogen. Hydrogen is a combination of hydrogen plus methane. In a single reactor system, we are regulating the pH, then we can produce methane as well as hydrogen. So similar to compressed natural gas, we can supplement up to 18% of hydrogen, which can be fueled into the existing engine infrastructure. So this also was approved by the government in the Gazette to use supplement less than 20% of hydrogen. So based on these inputs, we try to establish a refinery. This is the refinery platform which we have established with funding of CSIR to recover the products. The ones when we give waste or wastewater, initially it will go to a system where pre-treatment will be done to increase the organic carbon availability to the bacteria. Then it goes to the main reactor where 50% conversion of carbon will come into the hydrogen. And then we integrated with the third process where any of this product we can produce. So this is the closed view, top view of the biorefinery facility. This is the hydrogen reactor, blue one, where all accessories are there. Then we try to integrate with other unit operations so that carbon will be removed as well as resource recovery can happen. This is the top view. So here we try to look into the assessment, because green hydrogen, now Government of India with Ministry of New and Renewable Energy, the definition of green hydrogen is 2 kg of CO2 equivalents per kg of hydrogen production over a period of 12 months with the well-to-gate scenario. So we try to look into the analysis by life cycle analysis with standalone process where feeding will be food waste, a little bit of chemical, water and energy. This is standalone process where we are not considering the other recovery of byproduct. But refinery, we try to recover other products also. So when we look into the analysis, this is the about 16 impact categories. Specifically when we look into CO2 emissions, standalone process, I put negative because it's biogenic in origin, 4.21 kg CO2 equivalents per kg of hydrogen. Biorefinery because recovering all the carbon, so we are not leaving it to the environment, it's 1.203 kg of CO2 equivalent per kg of hydrogen. So this we can term, I cannot say it's a green hydrogen, but I can say it's a low carbon hydrogen. Unless we scale up to the certain level, we cannot comment on it. So damage categories also, when we looking to compare with the existing process, like M1, I didn't mention about the process, existing M1, M2, M3, M4, that's existing process in the market. So then all the impact is the human health, ecosystem, climate change, resources are relatively less when we went to the biorefinery process. This is a biorefinery. You can see the green one, where everywhere it is relatively compared to the less to the other process. So just we try to compare CO2 emissions from the different process which is available in market, tracking CH4 reforming, sodium amalgam diform, as well as electrolysis. So 1 kg of hydrogen, 1 kg of hydrogen production per kg of CO2 equivalents. When we look into electrolysis, the green hydrogen definition, we have to use the solar energy. So when we are using solar energy, it will be 2.7 kg CO2 per equivalent. However, when we are using the energy mix, which is coming from grid, it is 62.9 kg of CO2 equivalents per kg of hydrogen, which is not sustainable. So then we have a project with Dr. Atul, IIT Mandi. The objective is to produce hydrogen only. So hydrogen has a more application in the present existing system. So we try to use the waste and then establish a strategy, dual strategy, where we can produce methane as well as hydrogen parallelly and try to look into composition. So our objective is to use the food waste and vegetable waste in a decentralized way. Then this is the strategy. The first strategy is to increase hydrogen production, methane production, where pH will be 6.5 and a specific bacteria like acidogenic will be there. H2 plus CO4, that's a conventional methodic. So we can get H2, the hydrogen fraction, which is equivalent to the market available. So we try to look and optimize the conditions. We are able to get a significant amount of methane, which can directly go into the existing infrastructure. So the project has given us a very good leads. So that's what Atul also mentioned about biogenic hydrogen. So then at present we are looking into establishing demonstration plant. So earlier we have used the continuous mode operation. The conversion efficiency are less, but here we try to move towards continuous mode of operation, where per day we have to produce near to 1,50,000 liters of hydrogen per day using municipal waste. So in this process we have established a pre-hydrolysis. When we work with Dr. Atul, the major problem is availability of sugar. So we didn't put any pre-hydrolysis for wastewater. So we have established a pre-hydrolysis reactor where with less chemical input energy we get the sugars, which is a feedstock for hydrogen, and then we move to continuous mode operation and try to optimize flow rates, C/N ratio. Nitrogen is very important for bacteria to function. So by then we move to electrogenesis, like we put an electrode assembly into the system where it functions as a mediated electron transfer. So bacterial electron will be supplemented to the system again without losing, so that we can increase the performance. So this is the final results, where by inducing nitrogen, by inducing the organic loading rate optimum condition electrode assembly, we are able to increase hydrogen production from 194, the best condition, 208 to 260 liters per day in the system. So apart from increased fatty acid productions. So this is the block diagram, which in another 6 months we are establishing with funding from Department of Science and Technology. The module 1 is biological, where all feed preparation, pre-treatment and bioreactors are there. The module 2 is separation, pressure swing absorption, 3 stages pressure swing absorption to separate CO2 which is coming from the process. 55 to 60% is hydrogen, the remaining most of the fraction is CO2. So then the third module 2 is separation of fatty acids. So the fatty acids which is coming from aqueous waste, we are trying to separate and then treat the wastewater and use the wastewater in the loops. Those are the products which we are anticipating from this pilot plant. So that will reduce the cost significantly, hydrogen production cost, and also has a lot of environmental impact, positive environmental impact. So here again CO2 is a very critical thing. About 45% of gases emission is CO2. So we are trying to look CO2 as a feedstock. Basically microalgae is one of the potential thing which we can regularly convert CO2 and produce some of the bio-based product. But theoretically when we look into it, it's a slow process, required a lot of land. But recently people are working on bacteria, homocytogenic bacteria can take CO2, inorganic carbon, try to convert to some platform chemicals like acetic acid. So Lanzatech you might be knowing about, they are also trying with conversion of CO2 to ethanol with the engineered strain, but we are not going with engineered strain right now. We are using the strain. This is the strategy which we are going. The gaseous emissions coming from the acidogenic reactor, we will be looking to gas fermentation initially, but our interest is not gas fermentation. In gas fermentation, we want to introduce electrogenic system so that the input of hydrogen is not required. So CO2 to convert into some products, hydrogen is required as electron donor, but we want to eliminate that, and we try to supplement with electrons, with a little input of electrons, so that we can get the acetic acid, and then acetic acid is the product, and then we try to convert into ethanol. So this is the strategy we are working into. Tata Steels is sponsoring this project, this hybrid process. The basic thing is here we try to use the Woodlingle pathway. So Woodlingle pathway will try to convert 2 moles of CO2 in presence of 4 moles of hydrogen to acetic acid, but we are eliminating hydrogen there, 4 moles of hydrogen, and influxing the electron flux. So these are some of the results, gas fermentation. I will not go into deep. Consolidatedly, I try to present here some of the best conditions we got with the hybrid system, where we integrate electrodes as well as homoestrogenic bacteria. So this is a gas fermentation system which we are establishing now with that lead capacity for Tata Steel project to demonstrate the productivity. Further, once it is successful, we want to go for a higher scale at plant. So here one more thing I would like to mention. We are also looking into a different pathway where CO2 can be used to produce biosuccinic acid. So this is the metabolic pathway. Apart from giving glucose, when we supplement with CO2, there is a reverse TCA cycle, it goes. So reverse TCA cycle will give succinic acid. So this area also we are pursuing, because succinic acid is a very important bio-based chemical listed in 10 of the, in the 12 listed of US DOE. So we produce from biological route, again it's taking CO2, that's advantage. We have a strain, isolated strain, our own strain. We got very good productivity. Now we have established at 25 liter capacity, where we are getting 70 grams per liter of as succinic acid without genetic modification. So, we have to establish something, a refinery that should be self-sustainable by taking into advantage of photosynthesis initially to give the biomass, and then we can convert into different products, where hydrogen will be one of the products. So we can do it in a closed loop approach, taking into account of circular economy. Then we can reduce the CO2 emissions. So that's what, but lot of work has to be done in this area. Productivities are low, time of reaction is more, like near 20 hours in some cases. So if you are able to increase the productivity as well as conversion efficiency and reduce the cost with less rate intention, it can sustain. So as my students' work I have presented, funding, I would like to thank the funding which I got from different projects. Thank you for it.
Thank you so much, sir, for a detailed presentation. I think for a mechanical engineer like me, we would require more than a day to completely absorb whatever that you have said. But very briefly, there are a couple of questions. In terms of yield of biohydrogen coming from feedstock such as wastewater or for that matter any other feedstock, which is generally used for CBG production. So similar feedstocks, you say that it can be used for biohydrogen production. So typically, I just wanted to understand how much would be the yield of hydrogen for a ton of feedstock that we use. That is one thing in general, not necessarily based on your work. And secondly, on the commercial implementation side, do you think where we stand and what could be the major challenges, just for understanding?
The first thing is we are getting theoretical yield. When I look into 1 mole of glucose, 4 moles of hydrogen will be coming when we go to acetic acid pathway. But we cannot go with single pathway, it's a mixed pathway. So there we are able to gain 2.2 moles of hydrogen theoretically. So near to 55% of conversion. That's the maximum you got with the best condition. So remaining, because it increases, then there will be buffering problem. So activity will reduce, acetic acid, those acids will form. So even though it has capacity, cannot exceed that limit. So keeping that into consideration, we are looking, that's the maximum thing we have designed. So coming challenges, the major problem is the scaling of, we don't know how these bacteria will function at higher capacity. So 50 meter cube, we are able to control the process. But when we go with 3 times more, 30 meter cube, we are looking into. So how these bacteria behave? Can we sustain the estrogenic activity or suppress the methanolic activity throughout the operation? Those things has to be studied. That's why industry, if they want to demonstrate also, I am not going with industry. 30 meter capacity, we are able to demonstrate. Then we will have some leads to take up to. Other things are not a much difficult thing. So we want to use biomass, then pre-treatment additional step is required. But food waste from municipality, other things, not much pre-treatment is required. So that's what.
Thank you, sir. Thank you so much.
Just one point to be clarified that you mentioned about that.
It is okay.
You mentioned about that MSW to hydrogen. So that is excluding the plastic part, or it is only the biological part of the
Biogenic waste.
only the
Only
biogenic.
biogenic.
So excluded of that.
Yeah. So maybe municipal or food combination, composite one.
Yeah, hello sir. Nice. So actually I have a query regarding the uniform yields from the different type of waste. That is the main challenge. If you talk about the wastewater, if we have a steel industry, if we have another type of industry, we have a different composition of the wastewater. How you deal with that particular problem for the basically particular yield and uniform way to produce biohydrogen from these different composition of waste?
Yeah, that's absolute. So this depends on BOD by COD ratio. If it's more than 0.5, it's okay. But the system can sustain with respect to COD terms, organic carbon load. This system can function very effective up to 50 grams per liter of COD, whatever is the waste. So waste contain BOD will be 0.5. COD will be 50 grams per liter. That is, we can get the uniform hydrogen production. So we worked with different wastewater. This is the optimum conditions based on our studies.
Okay, thank you.
Thank you very much for the nice presentation. So when we are thinking about hydrogen production, the main idea that comes is electrolyzers. Maybe it's a wrong idea, but yeah, I was wondering, did you perform on those studies any economic analysis to assess about, for example, CapEx and OpEx and how it will compare with electrolyzers?
TEA I didn't do, but LCA, a little bit of LCA we have done to compare where we are standing. So TEA we have to do.
But you can give a cost around cost.
So for
Maybe what about the cost?
So
Compared
for hydrogen production with our process,
Yeah.
without separation we can produce to about $4. When we are using separation other products, we can come to $2.
Okay,
Two.
so that's
Two.
competitive.
Yeah.
That competitive.
Yeah.
Yeah.
Thank you.
Thank you, Professor. It's a very nice presentation and really a pity you don't have more time to talk about. I have one question. Regarding the core of the process, that is the bacteria, I would like to know if you have identified the precise species you are working with and what was the process to select them.
So basically,
Of course, it's biology and not chemical as a core.
it's a very important question. Initially, we tried with single cultures. So clostridium, Firmicute group of bacteria,
Yes.
they are known for hydrogen production. Basically, Clostridia, Bacillus. But when we are going with a mixed wastewater, we cannot add one more unit operation to make it autoclavable, like to kill all the bacteria. So if you are adding Bacillus and going with the process, Bacillus has to sustain. But for that, additional energy inputs are required. So we have adapted bacteria, mixed group of bacteria, and have a methodology to enrich estrogenic bacteria to maintain it. So group of bacteria will be in the system that will function in synergy. So that in small reactors we are able to maintain for 3 years without additionally giving any pre-treatment. But we have a strategy in between also. If we are losing the performance when we give diffused oxygen into the system, the anaerobic bacteria which are methane producing will kill. But these spore forming bacteria will not be there. So that also we have strategy.
Thank you very much.
Thank you.
Long live India and France friendship.